These are the phyla that we're going to be looking at over the next few lectures. And I'm going to start off with basically the ancient history of the animals and the group today that probably shares the most traits with this most recent common ancestor of all of the animals. That is the phylum Porifera, or the sponges. And it's the outgroup to the rest of the animal kingdom. The Porifera are quite curious because among all the animals, they are the least complicated and organized. There aren't even true tissue layers within sponge bodies. There's no body symmetry. So if you look at this animal, and yes, in fact, it is an animal, even though a lot of them were initially, or they were initially classified as some kind of plant, but you don't see any axis along which you can split it into two mirror image halves. Interestingly, the cells in a sponge retain a certain individuality about them. There are cells that do different things in a sponge, but they're not locked into those fates, what are called cell fates. They're kind of like stem cells in that they can de-differentiate and become something else. So any cell in a sponge can take on any role that any sponge cell can play. In addition, the cells of a sponge body are able to kind of make it on their own for a limited period of time. Being in this body and cooperating with other sponge cells is better for them than being out on their own. But they actually can survive on their own and, under certain conditions, reaggregate to reform a sponge that was taken apart down to the cellular level. That's weird. And it suggests that these organisms still today share a lot of the traits that the unicellular relatives of the animals may have had. Sponges do not have a whole lot of behavior that excites us, at least not on the macroscopic level. All of them are sessile. That means they don't move around. And they are all filter feeders. Filter feeding means that your food is very small particles, usually taken out of water. And there are a tremendous number of animals that make their living in this way in some fashion. They remove particles from moving water, and that provides them with all the nutrition they need. On the outside, they don't look like much. What you basically see is here is a big mass. You can see some little holes and some big ones. To see how a sponge works and what it's really made of, you need to get down at the cellular level. There is a type of cell called the choanocyte, or the collar cell, that is only found in sponges, and this is basically how the sponge makes its living, is with these cells. What these cells do is they generate a flow of water through the body of the sponge. And as that water moves through, any food particles that are floating in the water are filtered out, and that's what goes to keep the sponge fed. This is what a collar cell looks like. The collar are these microfibrils that are around one end of the cell. When these cells are in place within a sponge body, which is where you find them, what you have is the collar and this flagellum sticking out into the water column. And the rest of the cell body, is actually embedded in the body of the sponge. This hair-like structure, this is called a flagellum. You're probably familiar with that term from some encounters with microbiology in the past. But this is a structure that kind of waves back and forth like a whip. And that's what its job is in the collar cells. That generates a flow of water. So as the flagellum moves back and forth, it drives water through the collar of microfibrils, and any particles that are caught in that water pass through those microfibrils. Smaller ones are discarded, those just pass on through, but particles above a certain size are trapped and passed into the cell body, where they are digested intracellularly. This is a very individual way of feeding. So what this cell basically does is it feeds itself. There is no other digestive system in a sponge, although these cells can pass digested food, things like nutrients and sugars, onto other cells in the sponge body. Really, the independence of each one of those individual collar cells and the fact that they can sort of wander around, if necessary, independently of the sponge body, makes them seem very similar to a group of protists called choanoflagellates. And in fact, we found that choanoflagellates are genetically very similar to sponges. They are the outgroup to the animal kingdom. They're protists. They are colonial but unicellular, and they're found just outside of the animal clade. The strong suggestion there is that the sponges, and the first truly multicellular animals, therefore, actually evolved from a colonial choanoflagellate. Protists that originally lived independently, then gathered together into loose colonies, and that colony became more tightly organized until it evolved into something like a modern sponge. You may be wondering what this has to do with the sponge that you use in, say, your sink. And, well, frankly, the answer is that these are not real sponges. Neither is he. These are all sponges that are made of cellulose. That's where you get sponges today. They are not made of what is called spongin. The reason a sponge like this is called a sponge, like one of these, where's one? Here one, is because sponges that you use in the bath used to be the skeletal material of certain marine sponge animals. We don't do that anymore. Instead, we make our sponges out of things like plastic and cellulose and make Nickelodeon shows about them. You don't need to know the names of these three systems, but there are three different body shapes, basically, within the porifera. They're called the asconoid system, the cycanoid system, and the leuconoid system. And they're interesting because they show a theme of an organism becoming increasingly complex and efficient in a way that reflects some important truths about size and the complexity of transport. The asconoid system is the simplest one. In all these diagrams, the outer layer of the sponge is shown in yellow. The inner sort of medium, or the medial tissue, is shown in gray, and the red denotes the choanocytes, the collar cells that do the actual feeding. In the asconoid sponges, as these choanocytes set up a flow of water through the sponge, it pushes the water out of this large opening here. That causes water to flow in these smaller openings along the sides. These holes on the sides are called ostia. You don't need to know that. And as it passes through there, the water enters this hollow space, the spongiocele. The choanocytes, as the water flows through here, take up the food, and then the water that has been filtered is just ejected out of this large hole, which is called the osculum. This is a relatively inefficient way of feeding. And the reason is that you can see that most of the space inside the sponge is dead space. Remember, you can only catch food where choanocytes meet water. And here, the active feeding is restricted to this narrow area, this narrow layer of water that's around the outside of the spongiocele. Asconoid sponges cannot get very large because as they do, the surface area to volume ratio of the spongiocele drops. And so the sponge has more volume to feed and less area that it can use to feed its body with. So again, surface area to volume ratio comes back over and over again when you're looking at the way organisms are put together. There's a pretty easy change that you can make to an asconoid sponge to make it into a cycanoid sponge. Basically, what you do is you stretch out the body wall here so that instead of being a straight flat wall, it's all of these folds. So you take it and you change it into this. This is the cycanoid condition, or the cycanoid system. You've still got this big open space in the middle, but in order to get to that space, instead of passing through these little holes, water has to flow down essentially a hallway, all of which is lined with collar cells. That means that you've got much more surface area exposed to the water, and that means that this whole body can get a lot bigger and still feed itself. In an electron micrograph, this is what those fingers actually look like. You can see at this level of resolution, you can actually see these tiny little bumps here. Those are individual collar cells. And this is where the filtration actually happens in these little canals before you get to the spongiocele. A close-up of that shows the way that they're arranged. These are the pores. These triangular-looking things are pores through which the water comes. And then it circulates pretty slowly around in here, so the collar cells have plenty of time to get all of the food out of it. And truth be told, the cycanoid sponges are much better at getting food out of the water than asconoid sponges are. The next complexification, if you will, among the sponges is the leuconoid system. And in the leuconoid system, instead of having this big open space in the middle of the sponge, mostly what you have are just these canals that we saw first in the cycanoid system. They're canals leading in this almost a maze or labyrinth within the body of that sponge, and they connect these little round chambers that are lined with collar cells. So the water doesn't just go through one hallway and get filtered as it moves through the sponge. It can go through a whole web of hallways as it moves slowly into the interior of the sponge and then out a larger hole, which again is called an osculum. What happens as a result is that the sponge gets even more of a crack at the food that might be present in that water. It gets many more chances to get a hold of all of the nutrients that the water has to offer. And leuconoid sponges are by far the largest. They can get very large indeed, up to a couple of meters across. Here are what those individual chambers look like. They look remarkably like the alveoli, the little sacs in your lungs that are used for gas exchange. And lo and behold, this idea is the same. This is a structure having these little tiny round chambers is a structure that maximizes surface area. In this case of the sponge, the point of maximizing surface area is to maximize the amount of feeding. In your lungs, the purpose of alveoli is to maximize the surface area that's available for gas exchange. But the principle is basically the same. I mentioned earlier that the sponge body is unusually disorganized. You don't find anything that looks like a tissue, or certainly not an organ, within the sponge body. And all of the cells are capable of switching roles if they feel like it, or moving around. In fact, if you take a sponge body and you force it through a very, very fine screen so that it's actually physically broken up into individual cells, if you then just leave those cells lying at the bottom of a beaker of water, they will eventually start running into one another, and they will re-aggregate very slowly over the course of maybe a couple of weeks into a blob, and that blob will form a sponge. It will form a new sponge body. Okay, so you can break the individual cells apart and they will come back together and form the entire organism. Obviously, this is not something you can do with most organisms, like say a hamster. If you break the hamster down into its individual cells, what you'll have is a mess. In most animals, what happens is that once you get past a certain point in development, cells are kind of irreversibly dedicated to a particular identity, a particular task, and they can't go back again. They don't have the kind of individuality that you see in sponge cells. Sponges don't have a lot of the things that we associate with animals. They don't have any sense organs or nerve cells or electrical signals. They can in some way signal through their bodies. We're not exactly sure how. They do move a tiny bit to the extent of maybe opening some holes slightly and closing others slightly. So there is a certain amount of teamwork going on here. But there's nothing at all like a sensory or nervous system in a sponge.